Assalamu alaikum everyone. In this video lecture, we will be discussing the topical past papers of energy and respiration, which is a topic of A-level biology, or we can say A2 level biology. So this is the first question that we have to discuss. Table 8.1 shows some of the stages of glycolysis and respiration in anaerobic conditions in the yeast. These stages are not listed in the correct order. So guys, we have the stages written over here, and these stages are the stages of the glycolysis. And after the glycolysis, when the pyruvate is formed, the pyruvate is not entering into the mitochondrion. Instead, it is taking part in anaerobic respiration. And this is the anaerobic respiration in yeast, which is actually alcoholic fermentation. So these stages are not listed in correct order, and what we have to do that we have to place them in right order. Complete table 8.2 to show the correct order of the stages. One stage has been completed for you. Okay, so stage 4, or we can say, sorry, stage 5 is completed for us. Now we have to put these stages in correct order. Okay, let's do that. So these stages include glycolysis and alcoholic fermentation. So we will be starting with glycolysis. The first stage will be that of the glycolysis, when the glycolysis begins. So guys, glycolysis begins when the glucose is phosphorylated by ATP. We all know that glycolysis begins with glucose, and glucose is first phosphorylated by the ATP to form glucose 6 phosphate, right? So this is the first stage. Then what happens? After the glucose is fully phosphorylated, hexose bisphosphate is formed, or we can say fructose bisphosphate is formed. This is the second stage. Then what happens? As the fructose bisphosphate is formed, it splits, or it is lysed into two molecules of triose phosphates. The triose phosphates are then dehydrogenated by NAD to form reduced NAD. So this is the third stage. Then what happens when the triose phosphate is dehydrogenated? After that, GP is formed, and from the GP, pyruvate is formed. But before that, ATP is made by substrate linked phosphorylation. So this should be the fourth stage. And then the pyruvate should be formed, which is the fifth stage. Okay. When the pyruvate is formed, now what happens to the pyruvate? It is not entering into the mitochondrion. Instead, it is taking part in alcoholic fermentation, which occurs in the cytoplasm. So what will happen to the pyruvate? Because this is alcoholic fermentation, so pyruvate will be decarboxylated. And when the pyruvate is decarboxylated, what is formed? Ethanol is formed. Pyruvate is 3 carbon compound and ethanol is 2 carbon. So when the pyruvate is decarboxylated, ethanol is formed. And what happens then? This ethanol is then reduced by the enzyme alcohol dehydrogenase to form ethanol. So eventually ethanol is formed. So guys, these are the stages in the correct order. Let's put them in the table. So the first stage is E, and the second stage is H. The third stage is A. The fourth stage is I. The fifth stage is F, which is already written. The sixth stage is B. The seventh stage is C. The eighth stage is G. And the ninth stage is D. So we have placed the stages in the correct order. Let's move on to the next part. Part B states that yeast respires glucose in both aerobic and anaerobic conditions. Explain why less ATP is produced when yeast respires in anaerobic condition as compared to when the yeast respires in aerobic condition. So guys, you should know that in anaerobic conditions, only glycolysis occurs. So only glycolysis occurs in anaerobic conditions, right? Which results in the production of 2 ATP. So only glycolysis occurs in the anaerobic conditions, yielding 2 ATP, or we can say there is net production of 2 ATP. We also say that because oxygen is absent, so what happens? Oxygen is absent, so there is no final acceptor of electrons and protons. There is no final acceptor of electrons. As a result, what happens? As a result, the oxidative phosphorylation stops. As a result, the oxidative phosphorylation stops. And what happens because of that? The oxidative phosphorylation stops. And as we all know that most ATP are formed in oxidative phosphorylation. Most ATP are formed in oxidative phosphorylation. That's why, because oxidative phosphorylation is not occurring, so in anaerobic conditions less ATP are produced. Oxidative phosphorylation occurs in aerobic conditions when oxygen is present. We can also write that the ethanol that is produced still, it is energy rich, it is unable to break down further, so we can say pyruvate is converted to ethanol, which is still energy rich. Pyruvate does not enter the mitochondrion and is converted to, does not enter mitochondrion because oxygen is absent. Pyruvate does not enter mitochondrion and is converted to ethanol, which still contains energy, or which is still energy rich. Okay, so this is for the 5 marks. Okay, now we will move on to the next question. Okay, so let's discuss this question. Oxidative phosphorylation takes place in the mitochondrion. As we all know that oxidative phosphorylation is the last stage of the aerobic respiration and it takes place in the mitochondrion. Table 8.1 shows the different stages of oxidative phosphorylation. These stages are not listed in the correct order. So guys, table 8.1 shows all the stages of the oxidative phosphorylation, but these stages are not listed in the correct order. So what we have to do is we have to place these stages in the correct order. Complete table 8.2 to show the correct order of the stages. 2 of the stages have been completed for you. Okay, so let's place these stages in the correct order. If we discuss the oxidative phosphorylation, so from where does the oxidative phosphorylation starts? It starts when the reduced NAD, they go to the electron transport chain in the inner membrane of the mitochondrion and they release hydrogen atoms to the cytochrome carriers, which are actually the carriers of the electron transport chain. So this is the first stage. What happens then? The hydrogen atoms that are released by the reduced NAD, these hydrogen atoms then split into protons and electrons. So this is the next stage. Now what happens to the electrons? Electrons are picked up by the electron carriers or the cytochrome carriers, and these electrons are then passed from one carrier to the next or carrier to carrier. So this is the third stage. Then what happens that as these electrons are passing from one carrier to the next, energy is released, and this energy from the electron transfer is used to pump hydrogen ions or protons from the matrix to the intermembrane space. So this is the fourth stage, right? And because the protons or hydrogen ions are pumped from the matrix to the intermembrane space, a proton gradient or a hydrogen ion concentration gradient is set up across the crystal membrane, that is the membrane between the matrix and the intermembrane space. So this is the fifth stage. Then what happens is that protons then diffuse down the concentration gradient through the membrane proteins, or we can say ATP synthase, into the matrix, that is the sixth stage, that protons return back to the matrix by diffusion, and as the protons or hydrogen ions, they diffuse back into the matrix from the intermembrane space, energy is released, which is used to make ATP, and the enzyme that catalyzes the formation of ATP is the ATP synthase. So ATP synthase produces ATP. This is the next stage, seventh. Okay, so at last what happens is that the protons that come from the intermembrane space back to the matrix, these protons combine together with the electrons that are coming from the electron carriers. These protons and electrons, they combine with oxygen, which is the final acceptor of electrons and protons, to form water. This is the last stage. Okay, so these are the stages in the sequence. Now let's put these stages into the table. So first stage is V, second stage is S, third stage is U, fourth stage is W, fifth stage is R, sixth stage is Q, seventh stage is X, and last stage or eighth stage is T. Okay, let's move on to the next part, that is part B. State the precise location in a mitochondrion of each of the following. So guys, we have to state the locations of the mitochondrion for each of the following. So ATP synthase is located in the inner membrane or cristae. If we talk about the Krebs cycle, so you all know that the location of the Krebs cycle is the mitochondrial matrix. If we talk about the electron transport chain, then electron transport chain also takes place in the cristae or inner membrane, or we can say electron transport chain is found in the cristae or inner membrane. Coenzyme A, which takes part in the link reaction, is found in the matrix. If we talk about the pyruvate, so guys, pyruvate is found in the cytoplasm and the mitochondrial matrix, but in this question we have to state the location in the mitochondrion. So that will be the matrix, not the cytoplasm. Now we will move on to the next question. Figure 6.1 outlines some of the steps of glycolysis. So we can see the pathway of glycolysis in figure 6.1. Part 1. State the precise location of glycolysis in the cell. So as we all know that glycolysis occurs in the cytoplasm in a cell. So cytoplasm is the answer. Part 2. With reference to figure 6.1, state the steps where phosphorylation occurs. So guys, phosphorylation is the addition of a phosphate group. So we can see that in step 1, what happens? Glucose is converted to glucose 6-phosphate. It means phosphate is being added and phosphorylation occurs. The phosphate is added to the glucose. If we talk about step 2, then in step 2 what happens, that glucose 6-phosphate is converted to fructose 6-phosphate. There is basically change from glucose to fructose. There is isomerization. Fructose and glucose are isomers, so simply this reaction is isomerization. And there is no change in the number of phosphates, so phosphorylation does not occur in step 2. If we talk about step 3, then fructose 6-phosphate is converted to fructose 1,6-bisphosphate. So we can see that one more phosphate is being added in step 3. So guys, step 1 and step 3 are those steps where phosphorylation occurs. Okay, state the step where oxidation occurs. So guys, oxidation means dehydrogenation. So dehydrogenation is also known as oxidation. Dehydrogenation is the removal of hydrogens, and so is the oxidation. So we have the knowledge that whenever triose phosphate is converted to pyruvate, so during this step, triose phosphates are dehydrogenated or there is oxidation of triose phosphate. So in step 5, TP are oxidized or they are dehydrogenated. Name the type of reaction by which ATP is made during step 5. So guys, in step 5, we know that ATP are formed, and the ATP are formed by a type of reaction which is known as substrate linked phosphorylation. When ATP are made, they are either made by substrate linked phosphorylation or oxidative phosphorylation. So in glycolysis, substrate linked phosphorylation occurs and not the oxidative phosphorylation. Okay, part B. Some cancer cells have different metabolic requirements from normal cells. These cancer cells obtain most of their ATP from glycolysis even if oxygen is available. State how the glucose and oxygen requirements of these cancer cells differ from normal cells. So guys, we just have to state and not explain. We just have to give a statement or just a simple answer. So in this question, they're asking how the glucose and oxygen requirements of the cancer cells they differ from the normal cells, and we have the information at the top which says that cancer cells obtained most of their ATP by glycolysis and not by oxidative phosphorylation or aerobic respiration. Cancer cells obtain most ATP by glycolysis and not by aerobic respiration, or we can say oxidative phosphorylation. So because cancer cells are carrying out a lot of glycolysis, so they must be utilizing a lot of glucose. So we will say that glucose requirements of cancer cells will be higher. Glucose requirements of cancer cells will be higher as compared to the normal cells, whereas the oxygen requirement will be lower. We don't need to explain why, because the question says state. So oxygen requirements of cancer cells will be low because they're generating most ATP by glycolysis and very less ATP from the aerobic respiration and oxidative phosphorylation. Okay, let's move on to the next question. And what is that? Isolated mitochondria were used to investigate respiration. Okay, so we have isolated the mitochondria. We have isolated the mitochondria from the cells, and when we isolate the mitochondria from the cells, we put the mitochondria in buffer solution. Mitochondria were extracted from respiring mammalian cells and incubated in a buffer solution. As I just mentioned, that mitochondria are extracted from the mammalian cells, which were respiring mammalian cells, and they were kept in the buffer solution. Pyruvate and inorganic phosphate were added at time 0. ADP was added 1 minute later. The oxygen concentration of the buffer solution containing mitochondria was monitored throughout the investigation. So guys, we can see the graph, and in this graph on the y-axis there is oxygen concentration of the buffer solution that contains mitochondria. So oxygen concentration is being monitored at time 0. Pyruvate and inorganic phosphate are added. So you should know that when pyruvate is entered, or we can say pyruvate is inserted into the buffer solution together with inorganic phosphate. So what will be happening? Pyruvate will enter the mitochondria and pyruvate will take part in the link reaction, right, and the Krebs cycle, right. And we can see that ADP is added on 1 minute. Okay, so part 1 suggests why the line of the graph in figure 7.1 is steeper during phase B than during phase A. Okay, so they are asking the explanation why the line during the phase B is more steeper as compared to the line in the phase A. So we can see, guys, that in the phase A, oxygen concentration falls, but there's a very little fall in the oxygen concentration. But if we talk about phase B, there's a very sharp decrease in the oxygen concentration, right? Okay, so why is that so? This is the question exactly. That's because ADP are added at time 1 minute. This means that phase B contains ADP. And ADP is required for the synthesis of ATP. And if a lot of ATP are available, more ATP will be synthesized by oxidative phosphorylation. And in oxidative phosphorylation, oxygen is used as a final acceptor of electrons and protons. So more oxygen will be used, and oxygen rate of oxygen rate of fall of oxygen concentration will be higher, right? So what will be right over here? In phase B, ADP are present. In phase B, ADP are present. And ADP are required for what? ADP are required for ATP synthesis, because ADP combines with inorganic phosphate to make ATP. So ADP are required for ATP synthesis. Okay. So more ATP are synthesized by oxidative phosphorylation, and oxygen is the final acceptor of electrons, in oxidative phosphorylation. So more oxygen, or we can say, so rate of decrease of oxygen concentration is higher. Okay, let's move on to part 2. Suggest why the line of the graph starts to level out in phase C. Okay, we can see that the line levels out, almost levels out in phase C. That's because ADP are depleting, or we can say pyruvate or inorganic phosphate are depleting. We can just say that ADP or inorganic phosphate or pyruvate, they run out or they are depleting. That's why the graph in phase C levels off. Okay, let's move on to part B. Some plants, such as rice, grow with their roots submerged in water. Figure 7.2 shows a group of rice plants. Okay, as we all know, we have discussed that in the topic that we covered. The topic was energy and respiration, and at the end of the topic, I taught you guys that rice plants can survive in soils which have a lot of water, which have a lot of stagnant water, right? Rice plants can survive in flooded soils, or we can say that the rice plants, the roots of the rice plants, they are submerged in water, the soil is full of water. Explain how rice is adapted to grow with its roots submerged in water. So remember this point: when the soil contains a lot of water and the roots are submerged in water, so what happens is that the oxygen concentration is very low in the water, right? Oxygen is very slightly soluble in water. So remember this point, that oxygen concentration is usually higher in the air, but oxygen concentration is very low in the water. And because of this, less oxygen is supplied to the roots. But because of the other adaptations, the rice plant is getting a lot of oxygen through its several adaptations that we will be writing in this question. So we will say that usually, what we will say, that there is low concentration of oxygen in water, but the rice roots still get the oxygen because of the parenchyma. So guys, what is parenchyma? Parenchyma is simply a tissue which contains a lot of air spaces. We will say that parenchyma contains air spaces, or before that we can write that parenchyma is found in stems and roots, and it contains what? Air spaces through which oxygen reaches or oxygen diffuses down from the stems to the roots. So because of the parenchyma, the oxygen from the stems can reach to the roots, and roots can get sufficient oxygen for aerobic respiration. We will write so that roots can carry out aerobic respiration. What are the other adaptations? The leaves that are underwater. The leaves underwater store air in ridges or folds on the leaf surface. So we discussed that in the lectures that I covered. We discussed that the rice leaves which are underwater, they have ridges or folds on their surface which can store or trap air underwater. Okay, what else? The roots of rice plants contain high quantities of an enzyme called alcohol dehydrogenase, which can break down alcohol or ethanol. As a result, the roots can carry out anaerobic respiration at high rates, because roots can tolerate high concentrations of ethanol. We discussed this in our lectures that when the roots of the rice plants, they carry out anaerobic respiration, so the roots produce ethanol, but the roots contain an enzyme called alcohol dehydrogenase in high quantities, that's which this enzyme breaks down the ethanol, and because of this the roots of the rice plants can tolerate high concentrations of ethanol, and because of this the roots can carry out anaerobic respiration at high rates, producing a lot of ATP. Right. So guys, these are the adaptations of the rice plants to survive in waterlogged soils, or we can say with their roots submerged in water.